Okay, I want to thank everybody for joining us for today's webinar entitled The NSF Case Study Accelerating GC and MS Extractables and Leachables Analysis with Advanced Software Tools. Today's session is sponsored by Astrix and is going to be presented by the great folks at NSF International and Cerno Bioscience. We'll do those introductions for our speakers in just a moment. Before I get to the speakers, I just want to take a moment and introduce a little bit about Astrix. We can go to the next slide. And not sure, obviously, if you've those who are on the call with us today, if you've ever been on one of our webinars before. If you have not, and this is your first, I want to welcome you. If you are a returning visitor, we're certainly glad to have you back. So real quick, why is Astrix hosting these sessions? Well, first off, we are a consulting company and have been working exclusively in the life science space now for over 28 years. We bring a very unique blend of specialized services and technical consulting, as well as a very technology agnostic approach, meaning we work with lots of different vendors in the space, like the folks you're going to hear from today. What we're typically doing is helping folks kind of move their organizations and their R&D efforts of science-based organizations through digital transformation. 90% of the folks who work at Astrix hold advanced science degrees. We're extremely proud of that because we're a science-based company ourselves helping other science-based companies. So you'll be able to figure out how to learn more about Astrix when we get to the end of the session. With that, I want to take a moment now and introduce our speakers so we can go to the next slide. And before I turn it over to you, Ben, we have Benjamin Johnson at NSF. He's a manager of GC Bach Lab. And then we also have Yongdong Wang at Cerno Bioscience. He is the president and CEO. So we are thrilled to have both the folks with us today. Ben, I think you're going to take it first. So let me go ahead and pass it off to you, and I'll come back on at the end to help with questions.
Thanks, Kevin. Yes, I'm Ben Johnson. I'm a manager at NSF, and I'm here to talk about accelerating your GCMS analysis with advanced software tools. So first, a little bit about NSF. NSF was founded in 1944 at the University of Michigan. It was founded as a not-for-profit independent organization dedicated at the time to improving human health, but we've since expanded the scope to include improving planet health. Primarily, we do this by developing standards that are used to evaluate the safety of consumer products. And primarily, these products have been drinking water, but we also are involved with dietary supplements, food equipment, medical devices, and other things. We also offer certification to those standards and some other services, including consulting and training and education. When it comes to testing drinking water, we have two primary standards that we test to. The first being standard 60, which would cover any kind of drinking water additives that are used in purification of water. And then we have standard 61 that covers any kind of drinking water treatment equipment that might come in contact with drinking water. And we'll test products to both of these standards to ensure that they're safe for consumer use. And to do that, we test first for target compounds, which would be any compounds that we might expect to be present in the sample based on its formulation or its specifications. But we also test every product for non-target compounds, which would be anything else we might find in the sample. And these we'll try to identify, and if we can, try and provide a rough approximation of concentration of them. So this is a picture of the overall workflow, kind of how a sample comes through the lab. We'll prepare an analytical sample from the client sample. And with that sample, we'll acquire data on the instrument. We'll do target identification and quantification, followed by non-target identification and quantification. Then another analyst would review that and determine if the interpretations are correct, and finally we would generate a report to the LIMS system. So the problem that we had was with the non-target evaluation. The process that we had was entirely manual. It was slow, and it required highly trained chemists to do. And because of this, we began to accumulate a large backlog of samples, which was holding up our workflow. So at the time, we decided to start looking into ways we could automate some of these processes using software, and we developed some goals First being that we wanted it to be vendor independent. We're a global company, we have labs across the world, and we use different instruments, different software. We needed something that could provide the same evaluations no matter where it was being used. We also wanted it to be easy to learn and to automate as much as possible the sort of task elements of it. And we also wanted to decrease the expertise required. The chemists doing this are highly trained, and they're difficult to train and difficult to replace when they leave. So we wanted to find a solution that would allow people with less expertise to perform the same analyses. And finally, we wanted the interfacing with our LIMS system to be seamless. And at the same time, we started thinking that it might be a good idea to standardize our target analysis across labs. So we started looking for a chromatography data system that could work across our labs just as well. We had some requirements for that as well. Pretty standard CDS requirements, but we also wanted it to work with vendor neutral formats or open source formats like Andy rather than just proprietary data formats. We wanted it to be familiar and easy to train people on. And also we want something that can integrate with our LIMS system seamlessly without a lot of manual tasks involved. And so for our target analysis, we settled on thermochromelein as our desired CDS. It's sold independently of the instruments, and it can natively work with a lot of different vendors' instruments. Also, the interface is much like Excel, so the training is pretty minimal. It's fairly easy to learn. And it can accept the open source data format Andy. And using the Excel interface, there's powerful customizations available, so it made LIMS integration very simple. But one problem that we ran into is that proprietary MS data like Agilent or Thermo doesn't always integrate natively with Chromelein. So we needed a piece of software that could convert that to an open source format that we could import into Chromelein. And so we went to Cerno, and they developed a program that they call Candy, which can quickly convert mass spec data from any major vendor into an Andy format, which can be imported directly into Chromelein. I had some good features that make it convenient for lab use. And it also converts profile mode data back to centroid mode data. Profile mode data, as you'll see later on, was important for the non-target analysis, but target analysis is much more comfortable in centroid mode. So we wanted it to be able to transform the data back to centroid mode for the target analysis. Going back to the non-target analysis, I can talk a little bit more about some of the challenges we were facing. Every year we had 7,000 to 8,000 samples that had to be evaluated by a very small number of scientists. Everything about the process was manual. And sometimes some of the samples with more complex matrices could even take days or longer to evaluate. And even after identifying and quantifying all the peaks that were found in the sample, the review was tedious and manual as well. And then in order to integrate with our LIMS system, we were using sort of a legacy Excel macro that we had no ability to modify or maintain. So that was a pending issue as well. And as a result of all these challenges, the sample backlog could get up to 6 months at times. So when we were looking for a solution for this, we had some goals in mind. Primarily, we wanted to shorten the analysis turnaround time. But we also wanted to automate as many of the tasks involved as possible so that the analysts could spend more time making determinations and less time making manual tasks just to get the data to a format that could be evaluated. Again, we needed to be a vendor neutral format. And again, we wanted to reduce the expertise required for analysis, which would make more analysts available to do the work. And we had some other hopes that also we were able to realize here. And one is that we wanted to improve the confidence in the compound identification beyond what we were able to achieve with just NIST searches. And again, we wanted to integrate with our LIMS system. So we looked at several different software packages, but the one we finally settled on was Cerno's GCID. And the reason being, it sort of checked all the boxes that we're looking for. With vendor neutral, it came out of the box doing a lot of the automation that we were interested in. But it also had some interesting features. It had some really robust identification ability. And we can get into that later. But it also had the ability to provide all kinds of customizations for us, which really made it perfect for our application. And some of the customizations that we were looking for, we wanted it to be able to identify some of the known compounds in the samples and sort of identify or highlight them for the analysts so they wouldn't have to look at them and figure out what they were just to find out that they were an internal standard. We wanted it to do the quantitation for us. And we wanted it to annotate targets, surrogates, and other things that weren't needed in the non-target review, but were probably present in the sample. And then finally, we have established reporting limits. So we wanted it to filter out any peaks that were below that reporting limit before the analyst even had to look at it, so they wouldn't waste their time identifying things only to find out that they were not going to be reportable. And then we wanted to be able to make some manual assignments for things where we didn't believe the library searches were correct. So the final workflow with GCID looks like this. After the data is acquired on the instrument, we send the data in profile mode to GCID. It returns an evaluated spectrum annotated with all the peaks identified and match numbers assigned. The analyst can review the summary and determine which peaks seem like they need further evaluation. They can review those ticks, make the annotations, and then finally produce a LIMS report. And the GCID, or the GCID part of the process, is once the vendor data is imported, it automatically picks the peaks, does deconvolution, calculates the areas, performs a search on all the peaks that are found, and composites the search ranks, performs quantitation based on the nearest internal standard, and then it highlights all the peaks to indicate the quality of the match and the different types of known peaks that are present in the sample. So this slide is to start talking about the identification features of GCID. This is a standard sort of NIST search, and you can see the peaks for an unknown peak, or the possible identifications for an unknown peak, are basically listed by their forward search ranking. But for this case, you've got several possibilities, and the top match, being the Z isomer here, seems like it would be the likely candidate. But is it the correct one? In this case, it's not. In this case, the E isomer is actually the correct one, and GCID can show how this is the case. So when GCID does a library search, it combines the forward search, the reverse search, two additional match factors that we'll talk about later, as well as a retention index. And it combines all these factors into a composite match score, which allows you to determine that, in this case, the E isomer is the correct one. This slide, we'll turn it over to Yongdong to talk about.
Thank you, Ben. We come up with a approach to get more accurate information out of a conventional mass spectrometry system, such as the GC-MS, the single quad system that's being used here in this application. And the approach is a new way to calibrate mass spectrometry to make it more accurate, which is an elaborate process by itself, but when implemented on a GC-MS system like this, it can be done almost seamlessly, because we have readily available tune gas, the PFTBA tune gas that's available to us. We can acquire data from the tune gas to build the calibration, and then to be able to calibrate the raw data. In this case, is the black trace into the calibrated trace, the red trace, and in the process to improve the mass accuracy by a factor of 100, so that we can obtain accurate mass on a single quadruple or triple quadruple system without necessarily requiring a high resolution, more expensive system. And then in the process, we also obtain what we call the spectral accuracy, how accurate the whole curve is. And those two additional more accurate measurements can then be combined and implemented and integrated into GCID to generate confirmation and for the fragments and the molecular ion identified for the compounds that are found in the library. But in addition, if there's a compound that's not in the library, the accurate mass and spectral accuracy information can also help shed light on possible elemental composition, or even some information about the structures. So back to you, Ben.
Thanks. So a really powerful identification tool that's incorporated in GCID is the retention index calibration. The way this works is it can evaluate the retention times of well-identified peaks in your sample, maybe typically standard or something like that, and it can generate a retention index curve based on that. Or what we do in the lab is, before each run, we run a series of alkanes, which GCID can then identify and calibrate the retention index based off of that. Then once you have a calibrated GCID or GC, you can, any unknown peak that you detect from your sample can be assigned a retention index based on the calibrated standard. And as we saw on the previous slide, this is the primary factor for how it determined the difference between the E and Z isomers. The retention index fit were significantly different. So here's a typical scan of a not too bad sample after it's been processed through GCID. You can see, several colors in the assignments here. The greens indicates a known compound. The blue and yellow indicate the quality of the assignment. And this is what's really allowed us to reduce our evaluation time almost to a fraction of what it used to be. Each individual file can be processed in less than 4 seconds. You can process as many files at a time as you have processors on your computer. You get a match score for every peak in your sample. It quantitates them all based on the nearest internal standard. It also filters out all the peaks that are below our threshold. And it highlights and tags everything that is found with useful information to the analyst. And so you can see, after this file has been processed, the analyst now only has 2 peaks that they have to actually look at. And once they've determined whether these assignments are correct, they can easily export a LIMS file or a file to upload the data into our LIMS system. So in summary, we're very happy with the GCID solution. It's met all of our requirements for this project. It integrates with our LIMS system. It fully automates a lot of the tedious processes that we used to have to do manually. Our backlog is now down to nothing. Sample turnaround time for this analysis is roughly the same as for, a volatile scan or any other analysis that we're doing in the lab. Again, it's reduced the evaluation time from hours to minutes, days to hours, and it produces excellent LIMS reports, which keeps our data process simple. And that's all I have. Thank you.